Fantastic talk by Rajesh. I purposefully wanted this talk to be after him because this actually covers a lot of points that he mentioned. So how about I tell you that whatever he said, you could do it with a click of buttons, right? So introducing you the very first of a kind imaging-omic analysis, a robust AI-based software platform that answers 3 top questions. It gives user flexibility to control the parameters and do it in a more like a good user interactive way. B, it allows you to control your data at each and every point of your analysis. 3, it gives you a transparent AI report that allows you to track features through the entire algorithm, followed by the software. So let me introduce the field of radiogenomics. That was the inspiration to develop this software, first of all. So it basically starts with patients entering like the clinic where they get the MRI imaging done, radiologists segmenting the tumor, which basically gets biopsied and gets put on the slides, the tissue slides, in the pathology, where these tissue slides get stained and you get to know some molecular markers like estrogen and progesterone receptors in breast cancer, for example. And then these tumors get scraped off and sent to the genomics laboratory to do gene expression and next-generation sequencing analysis to get more detailed genomic profiles of these. What has been done so far is the texture features have been extracted from like texture, intensity, shape, and other imaging features have been extracted from these images and have been correlated with gene modules. So when I say gene modules, these are the cluster of genes outputted by these multiple genomic tests, and these are clustered based on the biological pathways. And they have been correlated with the imaging features. Further, moving forward from correlation, the ML modeling has been applied, different various kinds of models, as Rajesh pointed out, starting with linear regression, decision trees, random forest, the multi-layer neural network on the supervised learning data, etc. And then the efforts have been made to predict genomic data from the imaging features or vice versa, right? So that you could define the tumor heterogeneity better, that you don't just go from imaging to gene, but gene to imaging. So that was the inspiration for this kind of software to exist, because people were doing things differently in different labs. There was no standardization, like what model parameters should be used. Of course, they were doing it to achieve the best performance of the model, but then how about we find a way to track all of these and collaborate across multiple laboratories and sort of establish standard models that could be trained by you or me, or even evaluated further by Rajesh or anyone else sitting at any place in the world. So this software that I'm going to introduce is basically, I think I moved a slide forward, but so the idea of this software is to augment the existing software. So existing softwares have been Amaretto and Imaging Amaretto that does these kind of gene module to imaging feature correlations, and then it does the machine learning model part. You don't necessarily have to take the correlated features. You can take all the features to the machine learning model part, but it doesn't have a nice user like friendly web-based platform and doesn't go through each step in the algorithm to explain the matrices like AUCs, R-squares, RMSEs, and all sorts of matrices that you could think of, including feature importances that he pointed out. Like you want to know which features are important to train your model at each phase of your model training. And there's another software called Musa, which basically does all the statistical work but lacks the ML AI module. So there has been an unmet need in a broad imaging-omic domain where you need a sophisticated, user-friendly, and collaborative platform to analyze these data sets, build the model, and yield a descriptive report that could allow you to track through the features and gain a more understanding on the entire workflow. So here I introduce you this software called ImaGene Left side introduces you to the schema of ImaGene, which looks very simple, right? You have two data features files, right? Your imaging features or any other omic feature. It could be genomic, proteomic, patient outcome, therapy data, etc. You feed it into this software. It goes through some data normalizations, again, chosen by you through several methods that I'll be hinting in the next slides. I actually have a demo for you to present this software. So just bear with me for a moment. And then basically it goes to the correlation module, and then it goes to the ML module where you have multiple model types. These are just selected model types, but we have more model types when you see in the demo, and we are trying to augment it with more and more model types and test it on more and more cancer data sets. And then it gives you a nice descriptive report, just as how Rajesh mentioned. You want to track how your models are performing. You want to evaluate your data. You want to go back, feed it back to your model, and see how it's performing. And the good thing about this is this software is entirely hosted on Amazon Cloud at Queensland University. So you can perform multiple experiments in parallel. Like, you don't have to wait for one model to output the data. You can run 8 to 10 different models with thousands of data sets together in parallel. So that adds to the power of computing too in this kind of domain. On the right-hand side, it's a workflow. I don't mean to bother you with the details of it, but it goes through a chain of methods to evaluate the data at each point. And there's a feedback loop from the feature importances that could also be considered. And this software operates in two modes. The first mode does the correlations and the machine learning module, which does the modeling with or without the correlations. And the second part is the feature importance module, which does the machine learning modeling and spits out feature importance, feeds back the importance matrices for these features, and just select the features that could be retrained on the model. So you can go through this several times through even user intervention of modifying these data sets, how many zillion times you want to, until you arrive at the best performance of the model that you think will help you to advance your research further. So this tool is meant to steer your thoughts and processes and support researchers like you and me to do more advanced AI studies at your fingertips. So with that, I would like to just go through like a 10-minute demo, which will explain you the operation of the tool so that it becomes more clear, and then we'll go to the results of it. Yeah.
Hello, and welcome to ImaGene, a unique imaging-omic platform that allows you to correlate your imaging and omic features and build sophisticated ML/AI models off of it. You can create an account using Create Account button. This is a freely usable software hosted on the cloud of Queensland University of Technology. I'll start off by logging in into our lab's common Gmail account. Further, you can start the experiment by clicking on the upload file. Here, you can select from multiple model types, starting with linear regression, linear models, multitask lasso, decision trees, and more coming further in newer versions of ImaGene. So let's start with the linear regression. Any details regarding the linear regression model and the parameters used therein could be found here. This is a section where you enter model parameters. For example, any parameters that you could think in case of, let's select a linear model here. So we can use alpha, for example, 0.8, CV equal to 2, or you can keep it empty such that it uses the default parameters as the scikit-learn library uses in the link herein. You can select whether to do a grid search through hyperparameters, and the hyperparameters could be mentioned here in the form of going from certain values to the other, such that you could select a list of values for plethora of parameters that you wish to run your grid search through. For the sake of this tutorial, we'll just do with the grid search false. And here in the mode, you can select between different modes: train, predict, validate. We'll go with the training. Here you can select your test size. So 0.2 indicates out of the total data set, 20% of the data would be used for test and the remaining would be used for training. This is a k-fold cross-validation splitter parameter that you could indicate to cross-validate the trained data set within itself. Setting it to 2, we move further. For the data type and label type, you can select whether going from imaging to gene or gene to imaging. Normalize method could be selected either same or different for data and labels. We'll start with stand scalar. Correlation method could be selected either of Spearman or Pearson, and the correlation threshold could range between -1.0 to 1.0. Setting -1.0 allows you to completely silence the correlation module here, such that you take all the features into the AI module. We'll start with the setting the correlation filter threshold filter as 0.7. Essentially, this is a correlation coefficient threshold filter that allows you to filter through high correlations and take only those features through the machine learning module. Further, you can select whether to correct the p-value for your correlations. So I'll go with the famous Benjamin Hodgbuck. Next, we input data and label features here. So one thing to keep in mind is that your input file name should have underscores or hyphens, preferably underscores, and should not contain any spaces in them, such that the software could intake them appropriately and operate through to get you definitive results. So here we would select, since we indicated data type as imaging, we would select radiomic.csv as data and genefpkm.csv as our genomic feature file. And just to allude more how these files look like, these are essentially the comma separated values that could be opened up in Excel or could be prepped in Excel and stored as .csv, where they have a common ID column between both radiomic and the genomic or any other omic data file that you want to use. The features could be, genes or even patient outcomes that you want to train against. So one thing to make sure beforehand is the ID column is sorted in the correct order between the two files and should match exactly between the two files. This is the first test that the ImaGene performs. If that doesn't meet the criteria, then ImaGene will basically error out saying that IDs are not matching. So you would have to run them again. Taking it further from there, so once we have loaded the files, you have an option to choose whether you want to store the data for ImaGene, which is highly recommended, as this tool is meant to democratize radio-omic or imaging-omic correlations and machine learning model across the globe, such that a consensus could be arrived at these models and radiogenomic reports thereof to be shared across multiple labs worldwide. So I would highly request you to select yes. If you still think you don't want to share the data, you feel free to do no. Model file could be left empty because this is a training mode, where it would generate a model file for you. This option is to be used only when performing validation or prediction on the already cooked model. So here you could basically upload a pickle file, model.pkl, that you already generated through training of the model early. But for this tutorial, since we are going through the training mode, we will keep this empty. So once you submit and once everything gets accepted fine, it will give you an upload confirmation giving you the job ID, which could be tracked here as created, updated, and whether the task status is created and whether it's running or completed. As you could see that you could run multiple parallel experiments at the same time through ImaGene using the cloud power, such that you get results around the same time and you could compare through different models or perhaps different experiments between different features that you wish to. So for the sake of timeline here, I've already downloaded a report which was pre-cooked using the same approach. And so here, basically you would see two reports coming out of ImaGene. One is with the correlation and the ML module. Another one is without the correlation module and the feature selection mode, which automatically runs through ImaGene. So you don't have to worry about setting the feature selection mode here. Ideally, the report looks like this, where it gives you when the models were run, model inputs, multivariate correlations, the features selected post-correlation, imaging and gene features, what were the parameters that got finally used for model training, either with or without grid search, model evaluation for the trained data, just to peek into how the trained data look like, model evaluation for test data, where you have the scatter plots of actual versus predicted values for different omic labels that got predicted, the RMSE to standard deviation ratio indicating how well the model performed, some more matrices here giving you the observed mean and the ratio of RMSE to standard deviation, and finally the AUC values at different decision thresholds, aka the FPKM thresholds that were set here to predict the gene expressions at certain thresholds. So what happened was at each threshold, the data got binarized, the test variable got binarized, and it was evaluated whether how well it predicts the continuous gene expressions at each threshold using the imaging features, such that you could be confident that, for example, here at 0.5, the gene label ACTG2 gets predicted at a very high AUC value as opposed to 0.2, indicating that high expressions matter for this gene, versus some do go beyond 0.8 AUC for lower expressions. So this way you could connect the genes within the pathways post ImaGene runs and basically find the biological significance through looking through publications, whether other studies have reported the same, which we did, which is exactly the thing that we did in our manuscript. So please refer to that. Further, you have more validation data running permutations through each of these labels, only through the labels that have higher AUC, that is greater than 0.9, and R square greater than 0.25, such that you know whether this data is either randomly performing to be very accurate or it's actually confident with the p-values, with the strong p-values that we achieved, which were even like less than 0.002, such that it gives you confidence in the label predictions using the imaging features. Further, there are text files available in the output folder that gets downloaded from the ImaGene platform, where you could tally all the AUC results in the tabular format and work out your own plots if required. A nice wiki exists on our ImaGene's GitHub page, referenced on our manuscript. So it includes the entire flowchart of the ImaGene workflow that gets performed in the backend, and description of each of the functions used in the ImaGene scripts, starting with the Python main to the processing, to the normalization, building model, and evaluating it. Further, if you want to run ImaGene on a command line interface, you can download the ImaGene's Docker container from here and follow the instructions exactly to load the Docker container onto your Linux or Mac platform and run it as a command line interface exactly as shown here using the test data sets that we had attached here for radiomic, FPKM, CSV files, as well as some supplementary material for you to look at to check on the results. Thank you, and I hope you have a good time running ImaGene. If there are any questions, please forward it to me using my email ID on the authors list on the manuscript.
All right, so that was the demo of ImaGene, right? So now I'll dive into some results that we achieved using this software. So first use case is the invasive breast carcinoma, where the tumor infiltrates into rest of the breast tissue from the milk ducts or the lobules. As you could see on the MRI scans, it starts at one location and then starts spreading out, which basically signifies the term invasive here. So there were two aims that we want to achieve, and this time we did radioproteomics. So we wanted to predict the estrogen and progesterone receptor statuses from the radiology image features. So just to allude more to it, these are the receptor statuses that are deemed during the histopathology evaluation of the data. They take the biopsied material, they stain it, and based on the staining, and these are the very well-trained pathologists that are doing it, and all of the outcomes that they recorded, we have used that outcome. So there's nothing predicted from imaging features yet. When we used it, we of course wanted to sort it through like manually whether the ER and PR statuses exist from that, like for all the patients that we want to consider. So we tried to target it to the specific use case, invasive breast carcinoma, as more specific subset, more better outcome, especially in cancer. So we then tried to predict the ER and PR statuses from these radiology image features. What we then did was, whatever prediction we got, we merged them with the imaging features to explain the heterogeneity of the tumors on the radiology images, and we predicted the protein expressions in these tumors, which were measured by RPP assay by well-known institutes like MD Anderson. And the data set used here was from the well-renowned hospitals in the United States, such as MSKCC, Mayo, UPMC, and Roswell Park Cancer Institute. It's available freely on the Cancer Imaging Archive, TCIA, and the TCGA portals. We use the train and test. So basically, the data set was more focused because it was very well curated by radiologists. We only use the curated radiology features as opposed to just using all the features, and it was split at 95 to 5% between train test and validation. For the train test split was 80 to 20%, and we performed a threefold cross-validation within the training data set such that we could deem the performance of this model from the cross-validation point of view, which would indicate the overfitting of the model, which we did arrive at a nice score of 0.24. Then we compared that with the performance of test set and performance of validation set. We got a nice AUC for the ER status, 0.96, a mid to low performing AUC for the PR status with MSC altogether of 0.11. And then with the validation data set, we got a very tight performing MSC, which does correlate with the cross-validation matrix that we get that we got earlier. But we saw that the AUC for the ER status was right on spot. And again, these are the statuses outcome by the pathologists that we have used in the data. So they are like, they're trained for years to report out this directly to patients. So we would basically use only those data sets to do the predictions. However, the AUC failed for the PR status, and it is very well known in the invasive breast carcinoma that PR statuses are not that well prognostic compared to the ER status. So it kind of reflects in our data set here that ER prediction shows the high promise. Further, we combine the radiology and just the ER data to see if we could get to protein expressions and whether that would explain the heterogeneity in the patient tumors that we had. So we took the 71 very well focused data sets. We again split it into the percentages that you could see. And we this time try the multitask elastic net and lasso models, which basically means that they learn from each feature while they are trained. So if a feature A predicts feature B, and the feature C predicts feature D, first that feature learns from the first feature's prediction and tries to predict the other label better. So that's why they are called multitask. So the predicted proteins were, the top proteins are p-cadherin, which were predicted at the gene expression of low to mid, and EGFR was predicted as very low gene expression, and INPP4B was predicted at a very high gene expression, like 0.7 to 0.8 that you could see. And to deem the biological significance of it, we connected these genes in the pathway, and we found that EGFR is known to disrupt the E-cadherin protein, which is the buddy of p-cadherin, but p-cadherin comes into place when E-cadherin is running low. So EGFR disrupting the cadherins and EGFR being low expressed gives the room for p-cadherin to go up, and then INP44B protein is known to promote a pathway called WNT-catenin, which basically is found to be co-expressed with the p-cadherins. So it sort of helps us to connect dots between these three proteins post the gene prediction. So rather than making the gene modules earlier for the prediction, we did the single gene predictions, and then we connected them to the pathways, which would make sense for us. And especially for the p-cadherin part that you see, so these were the primary tumors that we used, which could be at their metastable, intermediate metastable state. So basically going from the epithelial to mesenchymal type, we found that the p-cadherin was performing low to mid, which means that these cancer cells are just in the EMT phase and could get converted to the mesenchymal phenotype anytime. So getting such good predictions and with the low to mid expression values, we were very confident that yes, it does have a biological significance, and we are now we are ready to validate this data set with much larger cohort from multiple hospitals and try to see if we could validate it really hard. So the study is underway. And another evidence of p-cadherin is it's known to classify the high grade and low grade histological subtypes of cancer. So basically, these high grades could be already in the EMT phase, the metastable phase, and the low grade could be just in the epithelial phase, waiting to be EMT driven. Another study that we performed was on the 195 breast cancer patients from Duke University. This time we focused on the therapy and therapy outcomes data and tried to predict the recurrence. Like we didn't have the data set on the recurrence sites, which we are in the process to collect that data from the collaborator there. But we knew whether in how many days did the disease recur after getting neoadjuvant therapy, adjuvant chemotherapy, and then through the doctor visits. So we had all these data sets. We found that the random forest model was really performing nice at AUC of 0.98 at p less than 0.002. And we are further in process to validate this data set using more and more data set from multiple collaborators. We have reached to AUC 0.65, but we are still evaluating which features are really important for our data sets. So this study is still under process, where you could see this feature importance graph that not all the features, for example, the therapy features compared to the tumor staging features that get evaluated in pathology would be less relevant. So the tumor staging features are getting the high scores. So this is the plot that you get from the data sets that you get from ImaGene, so that you could evaluate and then start minusing out the features, like just as Rajesh mentioned, or start including more features to see how the model performs. And then you can do rounds of experiments to see whether you get to the goal of achieving high performance for these kind of models. In conclusion, I would just like to say that ImaGene is the first kind of AI-based software that allows you to do imaging and omic, any kind of omic data analysis. It yields a transparent and descriptive ML AI report for users to track the features so that you could be very well accustomed to each step in the workflow. And it opens up the gates for collaboration across multiple laboratories to arrive at consensus models for prediction of multiple outcomes in cancer, which would basically aid precision medicine further. With that, I would like to acknowledge my PIs at Queensland Institute of Technology and Stanford, and our collaborators at IIT Delhi for their contributions. And thank you.